Hello, my name is Nicole Eaton, and I'm doing my model project on synaptic communication. Synaptic communication is between two nerve cells in order to pass electrical signal on to the next cell. A neuron is an excitable cell that sends information through different signals. Within the neuron, there is an axon, which conducts impulses and sends information to other neurons or muscles through propagation by jumping between the nodes of Ranvier. Extending from the axon, there are axon terminals, where they make synaptic contacts with other nerve cells. When an axon terminal or presynaptic neuron contacts a dendrite or postsynaptic neuron, a synapse occurs. The synaptic cleft is the space between the two neurons. Within the presynaptic neuron, there are synaptic vesicles carrying neurotransmitters through the intracellular fluid. In order for exocytosis to occur, calcium ions must enter through the L-type voltage-gated calcium ion channels, directing vesicles into the extracellular space. Once the neurotransmitters enter the synaptic cleft, they must go through the ligand-gated channels or ionotropic receptors to get to the postsynaptic neuron. The time it takes for a signal to travel from the presynaptic neuron to the postsynaptic neuron is known as synaptic delay. There are two types of neurotransmitters, which are excitatory and inhibitory. In excitatory neurotransmitters, sodium enters the cell, causing it to become more positive, which promotes action potentials. When the cell becomes more positive and reaches threshold at negative 55 millivolts, depolarization occurs, which means sodium channels are open. After depolarization, the sodium channels close and potassium channels open, causing repolarization. In inhibitory neurotransmitters, chloride enters the cell instead of sodium, making the cell more negative than it already was, causing hyperpolarization, making it harder to reach threshold and to create an action potential. And there you have it. That is how synaptic communication works.